Welcome to Muscle Tissue, Part 2. I'm Dr. Catherine Moore, the Histology Wizard. Today's short tutorial will cover the organization of smooth and cardiac muscle. We'll begin today talking about smooth muscle. In the last video, we covered some of the general characteristics of muscle, but smooth muscle is fundamentally different from skeletal muscle and cardiac muscle in terms of its structure, function, regulation of muscle contraction, and even excitation-contraction coupling. In this tutorial, we'll talk mainly about the differences in structure and organization. Most smooth muscle is mesenchymal in origin, although some muscle in the aorta and pulmonary arteries arises from ectomesenchyme, that is mesenchyme that's derived from neural crest. Smooth muscle is found in a number of different locations in the body, but it's predominantly found in the digestive system and in the vasculature and airways, and in a couple of the other areas listed here. And most of these are locations where slower, graded, and more prolonged contractions are important. For example, in the GI tract, the slower contractions help maintain a steady pressure on the contents. And in the blood vessels, smooth muscle regulates the luminal diameter of small arteries and arterioles, and this contributes to setting the level of blood pressure and blood flow to vascular beds. Now recall from the previous tutorial that smooth muscle, like cardiac muscle, is activated involuntarily, and that the action potential is transmitted via gap junctions, although again, the activation mechanisms are different. What does smooth muscle look like under the microscope? Well, smooth muscle cells are the smallest muscle cells, and they have a very characteristic spindle-like shape. And in contrast to the multinucleated skeletal muscle cells, smooth muscle fibers contain a single nucleus, often appearing wavy or shaped like a corkscrew or cigar. We'll talk about the reason for this shape in the video on muscle contraction. Note from this section showing smooth muscle in both a transverse and longitudinal orientation that the tissue is not striated, and importantly, for its ability to contract, smooth muscle fibers or myofibrils are not arranged in sarcomeres, and we'll come back to this point a lot. Smooth muscle is usually, but not always, found in two layers, and this most notably occurs in the digestive tube. In this cartoon of the small intestine, you can see the two layers clearly in this cross or transverse section through the organ. The outermost layer, or the longitudinal layer, regulates organ dilation and shortening, and in this image we can see the fibers of this longitudinal section actually appear in cross or transverse section. Sometimes this can be a bit counterintuitive, but if you refer back to the second image, the cartoon, you can see the longitudinal fibers on the outside wrapping the intestine. But you can also see that in this plane of section, not every nucleus is visible in every muscle fiber, and sometimes this makes it harder to identify. The inner layer is the circular layer, which controls organ constriction and elongation. Here we see fibers in longitudinal section, again a little counterintuitive, but here you can also see the shape of those corkscrew nuclei. Now, like skeletal muscle, smooth muscle can regenerate. Unlike skeletal muscle, which has limited regenerative abilities and relies on satellite or stem cells, every muscle fiber, every muscle cell of smooth muscle is capable of division when stimulated in response to trauma or injury. So smooth muscle actually gets bigger by hyperplasia, that is by cell division, versus cardiac muscle, for example, that gets bigger by hypertrophy. Now, smooth muscle cells are pretty cool cells. They don't just make muscle cells. They also secrete growth factors, renin, extracellular matrix components, and some of them can even migrate. So how can you tell the difference between smooth and skeletal muscle under the microscope? So first of all, it's important to remember location. If you recognize that you're in the digestive system, or you're looking at a blood vessel or an airway, you need to be thinking, hey, this is smooth muscle. Smooth muscle fibers can be pretty difficult to see at low mag, and this is compounded by the fact that you usually only see those nuclei in some of the cells. Now, one of the biggest clues about smooth muscle is that unless it's in a vessel or an airway, it's almost always seen in two orientations, because remember, it's found in two layers. Striated muscle fibers appear much more as individual units, and those skeletal muscles will show those multiple nuclei. Here I've summarized some of the more important features of smooth muscle for you to review later. We'll now move on to cardiac muscle. As its name suggests, cardiac muscle is found in only one location, the wall or the myometrium of the heart. Like skeletal muscle, cardiac muscle is striated, so it contains those same myofibrils and the same sarcomere arrangements as we see in skeletal muscle. However, cardiac fibers are shorter than skeletal muscle fibers. And as you can see here in this longitudinal section, this is in the top panel, cardiac muscle fibers often appear branched, and they often look like they have spaces in between, although that can be artifactual. This lower panel shows a transverse section through cardiac muscle, And here you can see that the nuclei for the most part are in a central position. You can also see in this section that cardiac muscle tissue is much less organized than skeletal muscle. So you don't see fascicles, nor do you see the connective tissue sheaths normally associated with skeletal muscle fibers. Overall, there is very little connective tissue in heart muscle, so you don't see paramecium or endomecium, for example. And overall, there's very, very little connective tissue in heart muscle, which makes a lot of sense given its function. Cardiac muscle tissue does have a lot of mitochondria, and this allows them to produce ATP very rapidly for contraction. So these cells can be fairly resistant to fatigue, which again is critical to keep the blood pumping. Cardiac muscle shares some similarities with smooth muscle. As I've mentioned before, both contain gap junctions that transmit an action potential for contraction. Both are activated involuntarily, although again, the mechanisms for activation and contraction are somewhat different. Finally, it's important to remember that cardiac muscle cells have the lowest ability to regenerate because they really don't contain stem cells. So there may be a few there, but not many. We're born with a set number of cardiomyocytes, and very few of these are replaced over our lifespan. This lack of replacement or repairability is one of the reasons why damaged cardiac tissue is usually replaced over time with connective tissue. This connective tissue, because it's not muscle tissue, lacks contractibility. That reduces the ability of the heart to pump blood. The heart can enlarge, so it can get bigger, and it does so both under physiological and pathological conditions. But this happens via hypertrophy, not cell division or hyperplasia. All heart muscle cells are connected by cellular bridges called intercalated discs, and these permit sodium, potassium, and calcium to diffuse from cell to cell easily. And this makes it easier for depolarization and repolarization in the myocardium. Now, because of these junctions, the heart muscle is able to act essentially as a single coordinated unit. Gap junctions have three major components. In this transmission electron micrograph image, you can see the desmosomes and fascia adherens junctions. So this is the Velcro again that holds these junctions together. You can also see the gap junctions that electrically couple the cells. In this longitudinal H&E section viewed under the light microscope, you can see these intercalated discs appear as dark lines joining the cardiac muscle fibers. So here's another short review, this time of cardiac muscle basics for you to review later. With this, we've now finished comparing the organization of the three muscle types, skeletal, smooth, and cardiac. And the next tutorial will tie together the structure of these muscles with their specific functions and compare the activation and mechanisms of contraction.